This tutorial will focus on what's understood by variable number tandem repeats, or VNTRs, or another name for this is short tandem repeats, STRs. The terminology is referring to the same concept. So basically, a short tandem repeat, or VNTR, is referring to a location on a chromosome where a particular nucleotide or base sequence is organised as what they call a tandem repeat. That is, a repeating sequence of bases, one after the other. These tandem repeats can be found on many different chromosomes and can often show variations. It's these variations that can act as alleles, that is, they can be variations on the same chromosome. And short tandem repeats are used particularly in looking at relationships in terms of paternity questions, or forensic analysis, or in other aspects of analysing genetics. These short tandem repeats are often divided into two categories. One category is referred to as microsatellites, which are usually nucleotide sequences of somewhere between 2 to 6 bases long, and it's these 2 to 6 bases that are repeated, so that forms the short tandem repeat, or mini satellites, which are often sequences of bases longer than 6, so anything up to 10 or more bases long in a repeating fashion. So mini just refers to a longer nucleotide sequence that gets repeated. Micro, somewhere between 2 to 6 bases long that gets repeated. So you might have along a particular chromosome, you might have a repeating base sequence of just 2 bases, say CA, 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 etc. On its homologous chromosome, that is the other copy of the chromosome that cells have, it may have the same, or it could be a different number of repeats. So if you have 2 different versions of the same segment, then you're talking about 2 different alleles. Or it could be that the sequences are the same. But the short tandem repeats refer to this repeating pattern. In this picture here, you can see this repeating pattern sitting within the length of a base sequence, and you can see that it starts here, CA, 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 and then it finishes there, and then it continues on with another series of bases. So here's your short tandem repeat, or microsatellite in this case. Another term that relates to the idea of short tandem repeats, or VNTRs, is something called restriction fragment length polymorphisms. Again, a terribly big word, sounds very complicated, but a relatively simple idea. The idea of a polymorphism relates to the idea that you can have two base sequences that vary slightly from each other. So it might be that on 2 chromosomes, so you might have, this might be chromosome number 1, and that might be the maternal chromosome, and chromosome number 1, and this is the paternal chromosome. So these are homologous chromosomes. So remember, we have 2 copies of every chromosome in our body. One set comes from our mother's side, and one set comes from our father's side, and these join in fertilisation. So we end up with 2 sets of every chromosome. So in a human being, we have 23 pairs of chromosomes. So 2 of chromosome 1, 2 of chromosome 2, 2 of chromosome 3, 2 of chromosome 4, etc., etc. So let's, for example, say this was our pair of chromosome 1s. It might be that we have a sequence that gets repeated in a section of the chromosome. Let's just say, for argument's sake, it's CAG, CAG, CAG. In this case, repeated 3 times. 1, 2, 3. Let's say on its homologous pair, we saw so we've got 1, 2, 3 repeats of CAG. In this one we have 2 repeats of CAG, and then we move into a different sequence. So on our homologous pair we have a polymorphism, that is 2 different versions of the same sequence. This one with 3 repeats and this one with just 2 repeats. So that's what we mean by a polymorphism. If they were exactly the same, then they wouldn't be polymorphic. They wouldn't have different variations. They would be the same. So in many sequences there are differences in the number of repeats, and this creates a polymorphism. The idea of a restriction fragment length polymorphism is in genetics you can utilise this idea by taking a DNA sample such as this and then cutting it with particular restriction enzymes which in some cases will cut out the required sequences, and then you can run it on gel electrophoresis, for example, to detect the relative lengths of the repeating sequences. So it might look something like this on a gel. So the wells of our gel are down here. We put our DNA fragments that we've cut from the previous example into the wells, and then we run the wells. So we run the gel electrophoresis. The DNA will move through the agarose gel for a set period of time, and we may end up with different banding patterns depending what's going on. So you'll remember, if you've looked at gel electrophoresis tutorial, that the further a DNA sample of DNA moves through the gel, the smaller are the fragments, or the shorter the fragments. The less distance they travel through, the longer or larger the fragments. So based on the previous example, if we go back for a minute, so this fragment here may be 9 base pairs long, for example, 3 repeats. This one here may only be 6 base pairs long, 2 repeats. So you might predict that this would be the 6 base pairs because it's smaller, therefore it travels further. This might be the 9 base pairs because it travels a less distance. So this is just a very generalised look at what restriction fragment length polymorphisms means and how it can be visualised. We can do some work in class that illustrates the actual application of this in real life, which might make a little bit more sense of it. But at least you have a little bit of an idea of what we're referring to here. This is true of any short tandem repeats, that they can sequence these repeats. They can work out exactly what the sequence of base pairs is and the number of repeats, and this in some cases can act as a prognosis for a genetic disease, the likelihood of getting a genetic disease, or it might look at where you've inherited that DNA from, so you might be able to work out who parents are. It might be able to provide a matching pattern on a gel to match, for example, a suspect for a crime scene and DNA at a crime scene, and the matching might give you evidence that a particular person was there.